My name is Demetra McILwain. My mentor is Dr. Randall Rasmussen in the Department of Biophysics and Physiology. My topic today is Timothy syndrome, the result of a single point mutation. I'm going to go over our research objective. Exactly why are we doing this research? Background information, what is Timothy syndrome, the Timothy syndrome mutation, characteristics of TS or Timothy syndrome, and the way it affects the heart, specifically how it causes long QT syndrome. I'll go over our scientific methods, what exactly we do in our lab, how far we have come, and where are we going with this research. Research objectives. To further gain information about Timothy syndrome in humans using a mouse model, first we have to establish and maintain the mouse model containing the TS mutation. Then we want to successfully genotype the TS mouse and characterize its phenotype. From there, we will compare the TS positive mice to negative mice and study characteristics of their tissues, specifically the heart, brain, colon, and uterus. Timothy syndrome is a rare autosomal dominant disorder in humans. It's the result of a single point mutation in the L-type calcium channel located on genes CAC and 1C on chromosome 12, this location here. It's found specifically on exon 8A, and there are about 50 cases in the U.S. The average death rate is 2.5 years. The TS mutation, as I mentioned, is found in exon 8A. Exons are just specific nucleic acids located on genes. And here you can see where the red G is highlighted. The name of the mutation specifically is glycine 406 arginine. And that's just implying that the glycine here is switched with an arginine, and that's why it's just a single point mutation. That one letter off causes this total disorder. Exon 8 at the bottom doesn't have the mutation. Here are characteristics of TS. It affects multiple organ systems, irregular neurodevelopment. About 80% of the cases of TS are related to autism, delayed social and motor development, and learning disabilities, cutaneous syndactyly of hands and feet. That's simply just the joining of skin. Usually you'll have 2 pointers or 3 middle fingers, 4 ring fingers, depending on how severe syndactyly is. There's facial abnormalities. Here you can see the low set ears, the flat nasal bridge, a round face, and these children here, they have hair, but usually baldness is a characteristic, along with the thin upper lip. Congenital heart defects are also caused by TS, and frequent infections due to a compromised immune system. TS cardiac manifestations, how it affects the heart. CAV 1.2 calcium channels are found, as I mentioned, in the heart, brain, colon, and uterine, but they're very prevalent in our heart. They're important for excitation and contraction of our heart muscle. So a mutation in this channel, the G406 arginine, will cause improper electrical conduction and structural changes of the heart. Long QT syndrome is the major cardiomyopathy associated with TS. It's simply a prolonged QT interval, the interval at which an electrical impulse is fired and the time it takes to recover. And it's prolonged due to a result of a large calcium influx going into the channel, decreasing the voltage dependent inactivation. In simple words, it takes much longer for the channel to close due to the increased calcium going in, which will obviously cause increase in heart rate, which is known as tachycardia, fibrillation, irregular heartbeats. You might feel you miss a beat, or your heart may stop for a while and restart. Arrhythmias, again, irregular heartbeat. Syncope, which is fainting, and overexertion. You're pumping so much blood to compensate for the structural changes that some people, their heart muscle hypertrophies would mean that it enlarges, which causes severe problems and can also lead to death. Sudden cardiac death is the leading cause in youth due to long QT syndrome. Here's an example of what a normal QT interval is. You can see about the length of the interval. Here's where it's prolonged. This ECG, or electrocardiogram, is often used as a diagnostic tool to diagnose long QT syndrome, but also to diagnose TS due to the high correlation. Other diagnostic tools include genetic screening. Scientific methods. What we do in our lab. We use genotyping techniques to simply identify the mutation in mice. Because there's such a low incidence of TS, we can't study the mutation in humans as of yet. And also due to the high mortality rates makes it difficult. So once we find the mutation, that's why making this mouse model is very important. So once we find the mutation in the TS mouse, we extract their RNA and convert it into cDNA. Using the cDNA, a portion of the calcium channel is amplified using PCR, and we can see the exon ratio, the exon A containing the mutation and the exon A which doesn't contain the mutation. Here's a PCR from our lab of the left ventricle and the heart, and these bands will indicate which exon is more prevalent, and the results haven't been published, so I can't really share them, but we have found that there may be more exon AA in the heart, which if there's more exon AA, of course, the higher risk of coming down with TS syndrome. So maintaining the TS mice mutation. We want to keep this mutation in our mice somewhat pure. It cannot be 100% pure, but we will try to get it as pure as possible to study the mutation clearly. We have a wild type mouse, and we'll think of the wild type as normal in nature, and it's the positive gene, the good gene, so to speak. And then there's the Timothy syndrome heterozygous. You have one good gene and one bad gene. If you have that bad gene, you will come down with TS. That's why it's an autosomal dominant disorder. And then if you have two negative genes, this is a fatal mutation. You get two negative genes from each parent, and these mice do not survive. Here's just a random, these are called Punnett square and Mendelian genetics. We have a heterozygous parent and a TS for TS and a wild type homozygous. You will see the results indicate that there's 50% of the offspring will come down with TS and 50% will be wild type. This is our ideal breeding scheme because we have our heterozygous and then we have our wild type so we can compare. These are two other breeding schemes that we don't really use because if we use all wild type homozygous, we don't get any of the mice with TS. And this at the bottom will produce 50% TS mice and 25% wild type, but it also gives us 25% of TS homozygous, the mutation which is fatal, and we don't want our mice to die. Here is what we have done so far. As I mentioned, just finding the mutation is a great achievement. So we found a way to PCR by taking DNA from these mice, and these are 7 mice, and we take DNA, sometimes by clipping their ears or clipping their tails to get the DNA, and we use a series of procedures to get the DNA in a form where we can extract it into a PCR. Here you can see a band at 1,200 base pairs. Base pairs is just the size of the DNA, and we use a DNA ladder to indicate the size. So if there is one band at 1,200, that means that the mouse is homozygous TS with the 2 negative genes, and those mice don't survive. If there are 2, one at 1,200 and one at 600, we call them transgenic, meaning they have the positive and the negative gene. One at 600 is our wild type, and again the 2 bands, which are heterozygous. Future research. Where are we going with this? We have found a way to use vivo morpholinos, which are compounds we inject into the mice, in hopes of suppressing the expression of the 8A mutation for TS. These studies are going on right now, and we haven't had that much luck, so we are trying to find a new method to inject the morpholinos. Further studies would include behavior studies of TS, for example, their maternal behaviors and social interactions, to help us understand the brain abnormalities associated with TS. And measuring voltage current relationships directly across cardiomyocytes. In our lab, we use frog oocytes to study currents across cell membranes, but at one point sooner than later, we want to use actual heart cells. But they are very, very small, about 10 microns in width, and very thin, and makes it difficult for us to hook electrodes up to the cell and to measure the voltage current relationships. The voltage current relationship will help us to further understand the effect that TS has on the heart and its electrical conduction system. Here are my references. And I'd like to thank those in the lab who have helped me, and CSTEP staff and interns.
When that box was moving, what it actually was like? I was just caught up about it was moving. Yeah.
Okay, at the top we have our wild type homozygous, the 2 positive. And then the bottom one shows a second parent, our TS heterozygous, the positive and negative gene. This is just a Punnett square which shows which gene will go into each box, will help us estimate the amount of offspring coming down with a specific genotype pattern. And the TS heterozygous will have the mutation, and the wild type homozygous will not have the mutation. Did that.
When you say you make the mouse, when you make a mouse model, do you use live mice?
Yes. There's a laboratory called Jackson Laboratory, and they specialize in making all these different type of mice. So we started off with a C57BL, it's just the name of a specific mouse. And we found a way to insert the mutation into that mouse, and then we would breed it with a wild type, and that's how we got with those patterns to produce more of the mice with the mutation in heterozygous. So we just keep breeding and keep breeding. And every day we go to the lab, we go and we check on these mice. And about every 2 weeks they have a litter of 12 pups. And if we can keep those pups alive, then that gives us more mice, and then it just keeps going from there. And so now we should have a pretty good breed going. But again, they will not be all pure due to mutations like this. You can't predict mutations.
So if they're positive, they have TS, and if they're positive, negative, they're carrier of TS?
The first one, the second one is right. But if they're positive, they're just wild type. They're what we would call the normal mouse. They don't have the mutation at all.
So what's the difference in lifespan between the wild type or the normal mice and the mice who had TS?
The normal mice would live a regular life if we maintain them properly, And the one with TS are the ones we take their tissues, so they don't live because we perform surgical procedures and extract those tissues of the brain, the uterus, colon, and the heart, of course. And those ones are sacrifice, so to speak, and we take those tissues to further study the exon ratios.
Okay, so you don't just allow them to live out to see differences.
Because they have TS, we have found that some of them die early, some of them die from unnatural causes. Sometimes the mother, there's a maternal instinct that say that a mother knows something is wrong with her child, and she may even eat her child, so they don't live all the time. So that's why maintaining this mouse model with the mutation is very important, because it's hard to keep up with these mice. And they also die for causes unknown, which we have to further study.
So sometimes certain types of diseases are prevalent in certain ethnicities. I know that you mentioned that there's a small group who have been diagnosed with TS. Have they been able to see that it is more prevalent in certain ethnicities?
Not as of